Replication process of prokaryotes. Starting with the initiation part, the initiation of replication requires a particular sequence called OREC, which is where the initiator protein, also known as DNA A, binds to the original replication. So let's say the original replication is right here, we bind right there. This causes a local unwinding in a short stretch of single-stranded DNA. Once it unwinds, DNA helicase comes in and attaches at the replication fork and moves into the fork, breaking these H bonds, as you can see right here. As the replication fork moves along the DNA, the then single-stranded binding proteins come in and coat the single-stranded DNA to protect it and stabilize it, as well as to prevent double-stranded DNA. A type of topoisomerase called DNA gyrase comes in and binds at both ends to prevent torsional strain. So it binds right here in order to prevent this part from supercoiling. Now we're at the elongation part, which is where an enzyme called primase comes and synthesizes short RNA primers to allow DNA synthesis. So they come through here. Then DNA polymerase 3 adds nucleotides to the primer and synthesizes DNA starting from the 3' to 3' hydroxyl group. So these 3 primes, so let's say this one's right here and this one's right here. Then the DNA polymerase 3 would come in and bind at those parts. And as it does that, it adds nucleotides to the primer. The strand grows antiparallel, which is where on the lagging strand here, the 5', it would then grow Okazaki fragments, which is these short fragments, incomplete fragments. And then on the leading strand, this 3' right here, it would grow a normal strand. DNA polymerase 1 comes in and then cuts out the RNA primer and uses the 5' to 3' polymerase activity to replace the RNA nucleotides with DNA nucleotides. So these parts that are incomplete are then taken care of by this DNA polymerase 1 and are able to convert to double strand right here. The DNA ligase comes in and seals the Nic. The Nic is the sugar and phosphate bond without adding another nucleotide to the strand, and that's pretty much it. Throughout the replication process, the parental DNA is used as a template in order to synthesize the leading and lagging strands. Thank you.